Good day everyone. We are the group 6 and this afternoon we are going to discuss about the genomic mapping and DNA sequencing. So first is, what is genomic mapping? Genomic mapping refers to the process of identifying the locations of genes or genetic markers along a chromosomes and determining the distance between them. So the genomic mapping is a one of a kind nga one of a best technique to determine or to find the the chromosomes and the genes. Then then it can provides a framework to understand the structures in the organizations of the genomes. So next is the history of genomic mapping. So year in 1991, Thomas Hunt Morgan and Alfred Stutterman studied fruit Drosophila melanogaster or servants created the first linkage map based on the crossing over frequencies. So in the first genetic map proposed, so si Thomas Hunt Morgan and Alfred Stuttervansky, siya ang naka first or siya ang nakauna-unang propose sa genetic map. So in year 1980s, um, allowed constructions of physical maps, detections of malformisms. Then restricted mapping RF markers developed. In 1990 to 2003, Human Genome Project is really comprehensive genome mapping and sequencing. So next is the importance of genomic mapping. So there are 5, which is the identifications of disease, variations and evolutionary studies, improvement of crop and livestock breeding, and advancement in personalized medicine. So first is the identifications of disease. So the identification of disease is the genomic mapping is it can help so scientists to locate the genes para to genes for responsible heredity like the disease, then by they're showing the specific mutations. Then the genomic mapping or the importance of the identification of disease is to give informations in the diagnostic can preventing a treating of genetic disorders. Then the second one is variations and evolutionary studies. So the genomic mapping reveals the genetic variations among the individual species. So the variations of evolutionary studies is naga-allow or the researchers allow to trace the evolutionary relationships and populations diversity. So the third one is the improvement of crop and livestock breeding. So the importance of the improvement of crop and livestock breeding in the genomic mapping is in agriculture the genomic mapping are it can be used to identify genes like uh, or like the desirable traits such as the disease resistance and the drought tolerance or the higher yield of the crops. Then Then the fourth one is the advancement and personalized medicine. So through the genomic mapping or the importance of the genomic mapping is it is possible to understand how individual genes affect their response to drugs. So it ano s- so the next is types of genomic mapping. So there are 3, which is the genetic or linkage map, physical map, and the last one is a sequence map. So the first type of genomic mapping is genetic linkage or map. So the first is the based on recombination frequency. frequency between genes shows on how often genes are inherited together. So, in the first type is it can be based on the recombination frequency between sa genes and the chromosomes, which is maka-inherit siya together. So, the second is physical map. So, based on actual physical distance between genes or DNA fragments in base pairs. So, in a physical map, it can be based on the physical distance, which is the distance between the DNA or the genes by its pairs or in base pairs. Um, the third one is a sequence map. Sequence map provides the exact nucleotides order of the DNA. This is the most detailed form of mapping. So the sequence map, which is the, if you say sequence is pasunod, which is to provide the exact nucleotides in order sa DNA. Then in the sequence map is dapat siya detailed form for the mapping. So the next one is components of genomic mapping. So the components of genomic mapping, there are 1, 2, 3, 4, 5. There are 5. First is genetic markers. Genetic markers, these are the identifiable DNA sequence whose inheritance can be tracked. So, in genetic markers, these are the to identify the DNA sequence, which is the sequences to for example is a parent, kung kinsa maka-inherit ato na genes, who can inherit those genes can be tracked. Then the second one is the recombinant or the crossover event. So during mitosis, homologous chromosomes can exchange segments or the crossover, which is causes recombination. So in a recombinant or the crossover event, so this have a possible between the homologous chromosomes it can be exchanged during the crossover during the crossover period, which is makakos siya og recombination. The third one is the genetic distance or map unit. Distance in genetic maps are often measured in centimorgans. It can be kanang ma-measured ang yaang distance, which is the distance between the DNA in the genome. Then the fourth one is the mapping functions. Because recombinant's frequency doesn't translate linearly into physical distance, or specifically when there are multiple crossover, mapping functions are used to convert recombinations data into map distance. So in mapping functions, it can be used if multiple crossover, physical distance in the multiple crossover siya, then the functions of the mapping is mag-convert siya to the recombinations into map distance. So bali siya ang measurement, ang mapping function. Ang mapping function is bali ang is siya ang measure sa the recommendations of the two. Then the last one is the linkage group and marker ordering. So marker that are linked or example inherited together more often than by chance are grouped into linkage group, which often corresponds to chromosomes or chromosome segments. So the the linkage group of and marker ordering is ano siya R group into linkage group, which is correspond siya into chromosomes or the chromosome segments. Then six is population and pedigrees. So the genomic mapping relies on having a suitable. So the genomic mapping to in a population is mag-rely siya, which is mag-base siya into the having a suitable in which recommendation events are observed and measured. Then the last one is application purpose, and genetic mapping is often when we use to locate the genes responsible for traits or disease by correlating markers and inheritance with the traits. So the application or the purpose of the genomic mapping is it can be used to locate or diba, to locate genes responsible for traits or kanang transfer siya traits or disease recruiting markers and inheritance with the trait. So major applications of genomic mapping. In the major application of genomic mapping. So first is disease gene identifications and medical research. So the genomic mapping, it can help to the scientists to locate the genes responsible for the genetic orders, which is kung mag-locate siya and to locate responsible genetic disorders. If kung gikan sa mag-locate siya og kanang responsible genes, then yang i-transfer into genetic disorders such as cystic fibrosis, muscular and dystrophy, and cancer. So second is the presentations and personalized medicine. So by understanding an individual genetic makeup through mapping, doctors can predict on how patients respond to specific drugs and create personalized treatment plan. So the third one is the agricultural improvement or plant and animal breeding. So genomic mappings allows to identify the genes on how to control the important traits such as the yield of the plants or the crops, then disease resistance and the drought tolerance. Next is the evolutionary and comparative genomics. So by comparing the genomes maps across the species or the scientists, across the species, so the scientists can study on how evolutionary relationships are genetic and genetic diversity work. Then next is the mark assisted selection or the MAS. So MAS, it can help plants breeders select plants carrying desirable traits. So MAS, which marker or selection to select the best traits or the best traits or genes, can help plant breeders para makaselect ng the best traits for the plant carrying into the desirable genes. So the next one is the identifying genes of core importance. So the genomic maps, it can help to locate controlling. So like for example, the yield on controlling on the yield, which is the yield of the plants or the crops, flowering time, nutrients use, efficiency, and stress tolerance. Next is the developing improved crops varieties. So the genomic mapping can help breeders to combine traits like sa higher yield and high productivity and the better quality of the crops or the fruits. So the next one is the studying genetic diversity. It can help scientists to understand what is or on how the different plants varieties are related together. So the next one is the step-by-step process. So first in the genomic mapping there are 1, 2, 3, 4, 5, 6. There are 6 steps. So first one is the first step is to collect and to collect and DNA sample. Ano to? Magkolekta and the DNA sample. Then the second step or the second step is identification and genetic markers. So after sa collection ng DNA sample, we are going to identify identify first and the genetic markers. Then the third, after the identification is we are going to measure the genetic linkage for the genetic maps. Then after the measuring genetics linkage, we are going to physical mapping. So mag-physical mapping na rin ta. We're going to use the physical mapping. Then after the physical mapping, so we're going to so the next is DNA sequencing and assembly. So after sa physical mapping, mag-ano din taog DNA sequencing and assembly. Then after sa DNA sequencing assembly, mo to ang map integrations. So next is we're going to discuss about the DNA sequencing. So first is, what is DNA sequencing? DNA sequencing is a process of determining the exact order of nucleotides or the adenine, AI, thymine, cytosine, and guanine in a DNA molecule. So the DNA sequencing is a process of determining exact copies. So kaning DNA sequencing, guys, is ano siya, is one of the process or is one of the best technique to process and to determine the exact order of the nucleotides. And it is a fundamental tools in the genetic and genomic use to study genes. So DNA sequencing is ano siya, is the one of the tools to identify the genetic and genomic on how we're going to use them, the genomic use to then on how we're going to use and how we're going to study about the genes and to detect mutations and to identify organisms and understand heredity disease. So that's all. Thank you.
History of DNA sequencing. 1970s, the beginning of DNA sequencing. The first DNA sequencing methods were developed in the mid-1970s. First, hay two major techniques: the Maxam-Walter and the Sanger method. The Maxam and Walter chemical degradation method, developed by Alan Maxam and Waller Gilbert, 1977, used in chemical modification and cleavage of DNA. So, ang nag-develop di ay kay si Alan Maxam and Walter Gilbert. Nag-use sila og chemicals para makat ang DNA and specific bases. Dangerous siya than complicated, so it's a rarely nga para siya magamit. Second, Sanger method, chain termination method, developed by Frederick Sanger, Necklin, and Coulson, 1977. Ang nag-develop di ay kay si Frederick Sanger, Necklin, and Coulson. Stop DNA change at specific bases to figure out the sequence, safer, easier, and more popular than Maxam Gilbert. In 1970s, DNA sequencing started. Maxam Gilbert, kay mo na siya ang chemical method, tricky and dangerous. Then si Sanger chain termination, easier and widely used. The first plant gene sequence. Scientists began sequencing chloroplast genes and mitochondrial genes in plant because these genomes are smaller than nuclear DNA. The first complete chloroplast genome, Tuba 1980 ko, was sequenced in 1986. So, they first focused on chloroplast genes and mitochondrial genes because these genomes are smaller and easier to study than the larger nuclear genome. In 1986, the first complete chloroplast genome was sequenced from tobacco. Chloroplast and mitochondrial DNA smaller and easier to read. 1986, first complete chloroplast genome from tobacco. 1990s, expansion to more plant species. PCR technology improved, allowing easier application of plant DNA. Research sequenced individual plant genes related to traits such as photosynthesis, disease resistance, and metabolism. In 1990s, more plant genes daw ang sequence para sa makaunsa sa better sa PCR. In early 2000, first complete plant genomes. In 2000, Arabidopsis thaliana became the first plant with fully sequenced nuclear genome. Is very important para sa mga agriculture. In 2005, next generation sequencing. New techniques, Elamina 454 solid-made sequencing of crop genomes accelerate includes corn, soybean, potato, and grapevine. So in 2005, technologies was introduced new methods like Lamina 454 and solid-made DNA sequence much faster, cheaper, and able to read more than A at once. Because of NGS, scientists could quickly sequence the genome of important crops such as corn, soybean, potato, and grapevine. In 2010, large-scale and plant genome. Scientists began sequencing thousands of plant varieties to understand genetic diversity. In 2010, DNA sequencing expanded to large-scale studies. Scientists began sequencing thousands of plant varieties to understand genetic diversity, difference in genes between plants. Plant genomes were created for crops like rice, maize, and wheat, showing genes present in some varieties but not others. Pangenome is shown which genes are present in some varieties but missing daw siya sa uban. Then, sequencing became a tool for breeding and identify genes. Breeding programs is to select and develop better plants. Then, ang identity genes is to find genes related to important traits. Importance of DNA sequencing. Identifying genes for important traits. DNA sequencing allows scientists to locate genes responsible for disease resistance, drought tolerance, high yield, and improved grain fruit or flower quality. Disease resistance helps plants fight off sickness. Drought tolerance helps plants survive with little water. High yield helps plants produce more food. Better quality of grains, fruits or flowers, bigger, fussier, or more attractive. Speed up plant breeding. Ang DNA markers is select good seedlings quickly. While ang DNA sequencing, kay ga-find siya sa plant disease. Helps improve crops and protect them from sickness. Types of DNA sequencing. Sanger sequencing, first generation sequencing. The first method used for sequencing DNA, accurate but slow and works best for small DNA fragments. Second, next generation sequencing. Fast, many sequences at once. Faster and cheaper than Sanger. Can read millions of DNA species at the same time. Commonly used for large-scale research and crop genomes. Ang third is third generation sequencing. The latest sequencing technology can read very long DNA sequence directly. Provides more complete information faster. Fourth, whole genome sequencing. Sequence the entire DNA of an organism gives a complete picture of all genes. Fifth, whole exome sequencing. Sequence only the cooling parts of genes, cheaper than WGS, and focuses on regions that make proteins. Number 6, target sequencing. Sequences specific genes of interest. Faster and cheaper when you only need certain genes. Lastly, 7, RNA sequencing or active genes. Sequence RNA instead of DNA. Show which genes are active in a cell or tissue. Useful for studying gene function and expression. Components of DNA sequencing. First is plant sample. Fresh young leaves are usually preferred because they have. So the first step in DNA sequencing is collecting a plant sample. Usually, fresh and young leaves are preferred because they have high DNA content and low secondary metabolites. So, ang high DNA content is more DNA to extract. And then, ang lower secondary metabolites is fewer chemicals that can interfere with DNA extraction. Other plants part can also be used, the roots, seeds, stems, and flowers. So, dapat ang plant sample is kanang young leaves siya are best for DNA sequencing. And other parts can work, but leaves are easiest and give the best DNA quality. This is the starting material for all the DNA sequencing. Last, DNA instruction regions. Plants' DNA extraction requires strong chemicals due to cellulose rich with walls, secondary metabolites, polyphenols, and polysaccharides. So, extracting DNA from plants needs strong chemicals because plants are different from animals. First is cellulose rich cell walls. Plant cells have those walls that are hard to break. Second, secondary metabolites, polyphenols, and polysaccharides. Chemicals like polyphenols and polysaccharides are in plants and interfere with DNA extraction.
Major application of DNA sequencing, genome sequencing, identifying genes, regulatory regions, and functional elements in plants' genomes. So means genome sequencing is like reading the entire instruction book of a plant. Inside this instruction book, you can find genes. Genes tell the plant what to do. Next is the regulatory regions. Act like an on-off switches, means the on-off is active and non-active genes. Next is the functional elements, help control growth, color, shape, and etc. Next is the crop improvement and breeding, or MAS, marker assistant selections. Means is a modern way to improve crops instead of waiting for plants to grow to look uh, grow grow. We look directly at the DNA. So we use markers or SN or SPNs. We use the SPNs as small flags in the DNA. These flags tell us which plants have good traits like high yields, disease resistance, good quality. Next is the identifying genes for stress tolerance, yield quality, and disease resistance. So means scientists also use genome data to find genes that help plants survive problems such as stress, low yield, poor quality, and next is disease. And the trait discovery is pinpointing genes responsible for drought and high tolerance, nutrient use efficiency. So the trait discovery is muna to pinpointing genes responsible for drought and heat tolerance. This means finding the genes that help plants survive with less water or in hot weather. Next is nutrients use efficiency. This is finding genes that help plants use fertilizer better. Next is the pest and disease resistance. It means this uh, these are traits that help plants fight pests and disease by themselves. Next, the flowering time, plant height, grain size, fruit color, and the flowering time. So the means these are visible traits that are controlled by specific genes. The flowering time is some plants flower early, some flower late, and the time in the plant height, a gene can make rice tall or short, and the grain is a gene can make rice grains long, short, or round. In the fruit colors is a gene make fruit red, yellow, purple, and etc. So next is we have the molecular identification and barcoding. Means this is using DNA to identify plants. It's like giving each plants a unique barcode, just like products in the grocery store. DNA barcodes help us know exactly what plants it is, even if they look similar. Next, some molecular identification and barcoding, we have the accurate species and identifications. It means the accurate species identification means DNA helps us confirm the exact species of a plant. The example of this is two herbal plants look almost the same. DNA barcoding can tell if it is lagundi or just a look-alike weed. Next is distinguishing varieties, hybrid, and cultivars. Means even if two plants look similar, then it can show which variety or hybrid they belong to. Next is detecting adulteration, adulteration in seed or plant-based products. Means DNA barcoding can check if a product or seed is pure or mixed with something else. Next is evolutionary and phylogenetic study. Means studying how plants are related to each other and how they change over time. Sa evolutionary and phylogenetic studies is na siya understanding plants evolution and speciation, meaning learning how one plant species slowly becomes a new species over many years. Next is comparing genomes across species to infer evolutionary relationships. Means comparing the DNA of different plants to see how similar or different they are. And the third one is tracking domestication paths or crops. Means finding out how wild plants became the crops we use today. And the next is transcription, omics, and gene expression analysis. Meaning studying which genes are turned on or turned off in a plant. So that means the turn on or turn off is is mo na ni siya ang atong active and non-active. Transcriptomics is like checking which plant's genes are awake and working. It is help us know what the plant is doing at the moment. Surviving stress, growing, flowering, or defending itself. The example is the draw stress. When a plant has no water, it needs to protect itself. So genes that help survives dry conditions are turned on. Gene for saving water, genes for growing deeper roots, genes for closing stomata. Transcriptomatics helps scientists see these on genes. Next is measure gene activity under stress or developmental stage. Developmental stage. So, scientists check which genes are turned on or genes are active or turned off, genes are not active, which a plant is stressed or growing. So, example, when corn has no water, when corn has no water, some genes are active on to help the plant survive the heat. Next is the identify alternative splicing and regulatory networks. Means a plant can cut and join one gene in different ways, making different proteins from the same genes. And scientists also study how genes work together and control each other. Next is the detection and mutation and genetic variants. We have the three. We have the under of the detection and mutation and genetic variants. First is the characterizing SPNs, insertion, deletions, and structural variants. So detection of mutation and genetic variants means finding any changes in a plant's DNA. Characterizing SPS, insertion, deletions, and structural variants means looking at what kind of DNA changes happened. These are different types of DNA changes. S and V, one letter in the DNA change. Ang insertion or deletions, something was added or removed. On the structural variants, a big change. Next is the mutation breeding. Mutation breeding means creating new traits by changing the DNA on purpose. Two common ways. EMS, the chemical that creates random mutations everywhere in the DNA. Scientists then search for useful changes. Then CRISPR gene editing cuts and edits specific gene. Very precise, like editing text in a document. And next is detection, mutation and screening for natural variation and germplasm collection. So, screening natural variations and germplasm collection means germplasm collections are libraries of many plant types from different regions. Scientists examine these plants to find natural differences that can help breeding. They check for disease resistance, growth tolerance, bigger seeds, better flavor, taste, growths. Then, step by step process. Number one is define the goal. So define the goal. So decide what you want to study or discover. Which example, which genes make this rice plant resistant to drought, and Next, number 2 is sample collection and storage. Collect plant parts, leaf, root, seed, and keep them fresh or frozen so the DNA does not get damaged. Example is take young leaves from rice, put them in a cool container or freezer. Next, a DNA extraction. Take the DNA out of the plant cells. The example is crush the leaf and chemicals and separate the pure DNA from the rest of the leaf materials. And number 4 is DNA quality control. Check if the DNA is clean, long enough, and not broken. Number 5 is library preparations. Prepare the DNA for sequencing by cutting it into small pieces and adding special tags so the machines can read it. Number 6 is sequencing run. So the sequencing machine read the DNA letters A, T, G, C. So the example is the machine produces millions of short DNA sequences from the leaf sample. And number 7 is base calling and raw data quality control. Is base calling, the computer converts the machine signals into DNA letters A, T, G, C. Error data, QC, check if the data is correct and not too noisy. Number 8 is read alignment or assembly. Two option alignment match the two options, which is they have the alignment and the assembly. Ang alignment is match the short DNA reads to a reference genome. Next, ang assembly put the short reads together to build a whole genome without a reference. So number 9 is polishing and scaffolding. Make means make the symbol genome cleaner, fix errors, and connect pieces into longer chromosomes like sequence. Number 10 is annotations. Find and label the gene, proteins, and important region in the genome. Number 11 is downstream analysis. So, means do deeper studies after annotations. And number 12 is validation and reportings. So, double check the results and write a report. Next is legal, ethical, and social issues. So the legal issues, we have the intellectual property rights, compliance with international agreements, biosafety regulation, data privacy, and ownership of genetic data. So the intellectual property or rights is means this law protect who owns a new discovery, invention, or genetic materials. It prevents other from copying of using the invention without permissions. Next is compliance with international agreements. So means countries must follow global rules when sharing genetic resources, seeds, or lab materials. And international agreement prevent unfair use of resources and protect countries from exploitations. Because these agreements ensure that genetic resources are shared fairly, benefits go to the right people, research is done responsibly. Next is the biosafety regulations. Means rules to ensure experiment involving gene, DNA, GMOs, and lab organisms are safe for people and the environment. So before releasing a GMO's crop like beauty corn, scientists must test if it is safe, check if it is harms other organisms, ensure it won't spread uncontrollably. Then these rules protect farms, communities, and the ecosystem. Next is the data privacy and ownership of genetic data. So genetic information or DNA data must be protected and controlled properly. We must decide who owns the data and how it can how it can be used. Genetic data be used. So because genetic data is very sensitive. It can reveal health, health, risk, identity, or ancestry. So it requires strict privacy rules. So next is the ethical issues. Ethical issues. Ang ethical issues is we have the fairness, access, and benefits sharing. Means so everyone who helps provide genetic resources or knowledge should get fair words. So dapat pantay-pantay din ang hatian sa benefits at access sa resources. Next, so manipulating plants' genomes means changing the genes of plants to make them better, like more resistant to disease or growth. And the next one, a biodiversity and environmental impact means research or GMOs should not harm other plants, animals, or ecosystems. Next is the consent for traditional knowledge. So before using the indigenous knowledge or plants from communities, their permission must be obtained. Example is if a tribe knows a plant cures fever, scientists must ask for permission before studying or patenting it. Next is the equity between rich and poor countries. So benefits from genetic resources or research should be shared fairly between countries. And next is the social issues. The social issues, we have the impact. First is impact on farmers. So means how new technologies, GMOs, or research affect farmers' lives. Next is public acceptance. So people must understand and accept the new technology or research. Next is the loss of indigenous crop varieties. So meaning traditional local plants are disappearing. So all local plants are lost. Next is the economic inequality. Meaning some people or countries get richer while others stay poor. Number 5, next is the food security concerns. Meaning is worry that people might not have enough safe and healthy food. So next is we have the biosafety. Biosafety, we have the laboratory biosafety during sample handling. So sa laboratory biosafety during sample handling, we have the safety handling of plants biosafety. Plants material, chemical safety. So laboratory biosafety is be very careful in the lab so that you and your team and the samples are safe. Why it matters? Some samples may carry bacteria, viruses, or other harmful materials. mishandling can cause illness or contamination. So, in simple words, be safe when touching lab samples. So, safety handling of plant materials means handle plants tissues carefully to avoid spreading pests, spreading pest, disease, or contamination. So, handle plants carefully so nothing gets sick or ruined. Next is chemical safety. Be careful with chemicals used in experiment to prevent accidents or injuries. Why? Because chemical can burn skin, cause poisoning, or damage the environment. So in simple words, use use chemical carefully to stay safe. So number 2 is biosafety during data processing. Biosafety during data processing. So means protect genetic information while analyzing it or computers. Why? Why it matters? Because DNA data can be sensitive. If it leaks, it could be misused. So keep DNA data safe on computers. So we have the under biosafety during data processing, which is the secure handling of genetic data. So DNA and genome information safety and only allow authorized people to access it. Why it matters? Because genetic data can reveal personal or sensitive information. Unauthorized access can harm individuals, farmers, or communities, so that keep DNA info private. Next is avoiding misuse of sequence data. So DNA or genetic info should not be used for harmful purposes. Why it matters? Misuse can lead to discrimination, unfair treatment, or environmental harm. In simple words, don't use DNA info to hurt people or nature. Next is the biosafety in downstream applications. So, after getting DNA results, be careful when applying them in experiments or agriculture. Why? Because improper use could affect humans, animals, or the environment. So, be careful when using DNA results. Next is genome editing and GM plant development. Making changes in plant chains like CRISPR must be done safely and responsibly. Why? Because if it's not done properly, GM crops could harm ecosystem or spread uncontrolled threats, so that makes new GM plants safety. Next is the preventing unintended release. So make sure genetically modified organism or GMOs or lab samples don't accidentally escape into nature, because if they escape, they could mix with wild plants, then spread uncontrolled threats or harms other crops. So stop modified plants or samples from escaping by accident. Next, the ecological biosafety. So protect the environment which when doing research or using GM crops, because some modified plants or insects can harm others, animals, plant, or soil. So don't hurt the environment or animals. Next is the biosafety regulations and compliance. So follow the safety rules for labs, experiments, and planting GMOs. Why? So why it matters? So prevent accidents, contamination, and unsafe experiments. So in simple words, follow safety rules in labs and fields. Next is the national laws. National laws meaning obey your country's rules for genetic research, GM crops, and plants protection. Why it matters? Because different countries have specific laws to protect farmers, consumer, and the environment. So follow the law of your own country. Next is the international agreement. Means follow global rules when sharing plants, seeds, or genetic resources with other countries. Why? Because ensures fairness and prevents misuse of genetic resources between countries. So you need to follow world rules when sharing plants or when sharing plants or genes. The number 5 is ethical and societal biosafety considerations. Meaning think about what is right or wrong for people's communities and society when doing research. Why it matters? Because protects communities, farmers, and indigenous people from exploitation or human. So do what is fair and safe for everyone. Next is the protecting the under of ethical and societal biosafety consideration is the first is protecting indigenous plant resource. Meaning preserves local and traditional plant varieties so they are not lost or misused. Why? Why it matters? Because heritage be- because losing indigenous crops reduce biodiversity and cultural heritage. So save old local plants and don't misuse them. And the next is preventing monoculture dependence, meaning avoid growing only one type of crop everywhere. Why it matters? Because monoculture increase risk of pest disease and crop failure, which can hurt farmers and food supplies. Don't grow just one kind of crop all the time. Ne- all the time. Next is the thank you presenters. Mamita.